Okay, so I'm going to share the testing process to detect the coronavirus by using the real-time PCR. Real-time PCR is a nuclear-derived method for detecting the presence of specific genetic material from any pathogen, including a virus. Originally, the method used radioactive isotope markers to detect targeted genetic materials. Uh, but subsequent refining has led to the replacement of the isotopic labeling with specific markers, most frequently fluorescent dyes. So coronavirus only contain RNA, which means they rely on healthy cells to multiply and survive. So, in order to detect the coronavirus in the body using real time PCR, scientists need to convert the RNA to DNA, and this is the process called reverse transcription. They do this because only DNA can be copied or amplified, which is a key part of the real-time PCR process for detecting viruses. And a sample is collected from parts of the body where the coronavirus gathers, such as the person's nose or throat. The sample is treated with several chemical solutions that remove substances such as proteins and fats and extracts only the RNA present in the sample. So this extracted RNA is a mix of a person's own genetic material and it presents the coronavirus RNA. So the RNA is reverse transcribed to DNA using a specific enzyme. And then the scientists then add additional short fragments of DNA that are complementary to specific parts of the transcribed viral DNA. So these fragments attach themselves to target sections of the viral DNA if the virus is present in a sample. So some of the added genetic fragments are for building DNA strands during amplification, while the others are for building the DNA and adding marker labels to the strands, which are then used to detect the virus. And then the mixture is then placed in a RT-PCR machine. It's real-time PCR machine. The machine cycles through temperatures that heat and cool the mixture to trigger specific chemical reactions that create new identical copies of the target sections of the viral DNA. So the cycle repeats over and over to continue copying the target sections of viral DNA. And each cycle doubles the previous amount. So that means 2 copies become 4, 4 copies become 8, and so on. So a standard real-time PCR setup usually go through 35 cycles, which means that by the end of the process, around 35 billion new copies of the sections of viral DNA are created from each strand of the virus present in the sample. As new copies of the viral sections are built, the marker labels attached to the DNA strands and then release a fluorescent dye, which is measured by the machine's computer and presented in real time on the screen. The computer tracks the amount of the fluorescence in the sample after each cycle. When the amount goes over a certain level of fluorescence, this confirms that the virus is present. Scientists also monitor how many cycles it takes to reach this level in order to estimate the severity of the infection. The fewer the cycles, the more severe the viral infection is. So the real-time RT-PCR technique is highly sensitive and specific and can deliver a reliable diagnosis as fast as 3 hours. Though usually laboratories take on average between 6 to 8 hours compared to other available virus isolation methods, real-time PCR is significantly faster and has a lower potential for contamination or errors as the entire process can be done within a closed tube. So I think all of these are the testing process to detect the coronavirus.